Thanks, Richard. So firstly, let me thank you and the University of Reading for giving me the opportunity to come and talk about some of our work, some of the work from our lab here tonight. As mentioned, I'll be talking about how we can understand depression better by looking at the neurobiology of depression. And I'll talk through firstly what we think about when we talk about the symptoms of depression, how we can understand the symptoms in relation to brain activity, and how some of the current pharmacological treatments that we have for depression interact with the brain's activity. Oh, slightly fast. So when we think of depression, we often think of the sadness and the low mood. And it's quite well known now that this symptom of depression seems to be maintained by what's called a negative bias. And what is a negative bias? This is the ability to really focus on negative information and an over-processing of negative information. So for example, when you can see the situation or the cup is half empty rather than half full, and it's mostly described, patients can only see the negative situations both in relation to themselves and to their experiences and their environment. And we have quite a bit of information now, well documented, about how we process negative information in the human brain. You may have heard of the amygdala. Amygdala is one of the oldest parts of the human brain, and this is part of the brain that seems to be active when we experience fearful or threatening situations. And we also know now from studies using neuroimaging, so techniques to look inside the human brain, that patients with depression have increased activity in places like the amygdala when they process negative information. And in fact, some of the pharmacological treatments that we currently have for depression target serotonin, and serotonin is one of the main chemical transmitters in the brain. That is one of the chemicals that translates information between neurons about how we process negative information, and this is one of the targets that we have with pharmacological treatments that currently are used to treat depression. Not only pharmacological treatments, but psychological treatments that we have today to treat depression seem to target how we process negative information. And that's all very well, but unfortunately the treatments that we have for depression aren't terribly good. They work for some people, and they work for some people very well, but they don't work for everybody. And in fact, the efficacy is really only between about 30 and 50%. It's not necessarily that psychological treatments are better than pharmacological. In fact, we can't really at this stage figure out who responds to psychological treatment better than pharmacological treatment, and with them even combined, the effectiveness isn't really any more than about 30 to 50%. One of the reasons for this is that we think that the treatments, both psychological and pharmacological, aren't really targeting all the symptoms that we have in depression. For example, I'm quite interested in anhedonia. Anhedonia is the second of the two main diagnostic criteria for depression. So if you present to the GP with feelings of depression, you have to meet certain criteria, and anhedonia is one of the key symptoms. So what is anhedonia? Anhedonia is this inability to experience reward and pleasure in what you used to find rewarding and pleasurable before the onset of depression. It's very nicely exemplified by many different authors and in many different writings, but I like this one by William Starren, the author, who wrote, "I find myself eating, if only for subsistence. Food, like everything else within the scope of sensation, was utterly without savour." And this is just a reminder of the types of symptoms that we're talking about when we're thinking about depression, this inability to experience pleasure and reward. So when we think about anhedonia, what do we think? It's possibly got something to do with how the brain responds to reward, right? It's got to be something to do with how our brains respond to pleasurable information, and maybe that's gone wrong, or there's some problem with that underlying anhedonia, the symptom of problems with reward in depressed patients. So how do we measure the brain's response to reward? Well, what we can do with the advent of neuroimaging, which has really only been around for about 10-15 years, we've been able to look inside the human brain. We know a lot about what goes on in the reward systems in animals, but with the advent of imaging, we can start to look inside humans non-invasively. So we can put people into a scanner and we can look at how their brains respond to reward. So on the left, this is actually the scanner that we have here at Reading University. And on the right, this is the typical structural scan that you get from an MRI machine. As you can see, it's quite detailed and it shows us up all the different parts inside the brain. But if we want to look at how people respond to information processing, so for example, how they process reward or how they process negative information, then we usually give people a task or different stimuli inside the scanner. And we call this functional magnetic resonance imaging. The function just means they're doing a task inside the scanner. And this is the type of data that we get from the scanner. So we can collect many, many images in about 30 minutes of a task. So for example, we collect approximately 3,500 of these images in a 30-minute test scan. Then we combine these images and we start to look at what parts of the brain are being activated or involved when the person is carrying out the particular task that we're interested in. So how does MRI work, or fMRI work? Well, this is a really simplified version. And basically, how it works is that fMRI, or the machine itself, is a big magnet. And it detects changes in blood flow. Now, the blood itself is magnetically charged. And when you're involved in a task or when you're processing information, we assume that parts of the brain that are needed to be involved in that task need more blood because they need more energy. So blood flows to the region that's involved in the task, involved in the processing of information. And basically that's how it works. So the magnet itself can detect the changes in blood flow. Using this, and also, as I mentioned already, the preclinical work, so all the work that's already been done in animals, we know now the different particular neurotransmitters that are involved in the processing of rewarding and pleasurable information. The one that jumps to most people's mind is dopamine. You've probably all heard about it before. It makes the headlines quite a bit. It's not the only neurotransmitter involved in reward processing, but it's one of the main neurotransmitters. And it's thought to be involved in how we are motivated to seek out rewards, and also in our experiences of actually feeling the pleasure once we receive a reward. And this is just some, don't worry about the particular brain regions, but this is just to highlight where dopamine begins in the brain, and all the different parts of the brain it can innervate. So it can reach different parts of the brain that we know to be involved in decision making, goal-orientated behaviour, pleasurable experiences, etc. So that's just a picture to show you how the different neurotransmitters are involved in lots of different parts of the brain, and these are the targets that we have for some of these psychiatric disorders. Like I said, it hasn't been that long that people have started to look at how we process rewarding information in the human brain. Most of the work's been done in animals, but in about the last 10 years, so from about 2005, people started to look at how depressed patients respond to rewarding information in their brain. And what they found basically is that as you have experiences of anhedonia, so the more experiences you have of feeling a lack of pleasure, so the less able you are to experience reward, the less brain activity you have in reward centers in your brain if you're depressed. And this is what's shown here. This is what's called a negative correlation. And all it really means is that as the symptoms of depression or anhedonia in depression, the symptoms of an inability to experience pleasure increase, the brain activity in key parts of the brain, like the ventral striatum, actually decrease. So this is well documented now in depressed patients. But anhedonia, so this inability to experience pleasure and reward, has been proposed as a possible biomarker for depression. What is a biomarker, you're probably thinking? A biomarker is a vulnerability or a risk factor, and for us they're really, really important. They're really the holy grail in psychiatry. It's the kind of thing that we're all after. Why? Because if we can identify risk factors, perhaps, and only perhaps, we could start to actually intervene early with people to try and give them treatments or preventative strategies before they ever actually go on to experience depression. And that would be really, really wonderful. So that's one of the reasons that we're interested in trying to find biomarkers. So this feeling of anhedonia, this inability to experience pleasure, is thought of as a possible biomarker. Why? Because it predates the onset of depression. It seems to hang around when you're depressed, and it doesn't go away when you've become recovered. So recovery, i.e. you no longer feel sad or very anxious in your depression, but you still have these feelings of anhedonia. So what we're interested in our lab is we want to know these feelings of anhedonia that seem to be a biomarker, something valuable, something worth finding out, something worth chasing. Are there markers of this feeling in the brain? Can we detect differences in the brain's responses to reward in people who are at risk of depression? And if so, this is another tick in the box for saying that reward function in the brain is a possible biomarker for depression. So this is the crux of the first half of my talk. This is the kind of thing that we're interested in finding out. So what are the risk factors for depression? Well, of course, this list isn't exhaustive, but these are some of the key things that we know to increase your risk of depression. One of the biggest risk factors for depression, and it might seem obvious to you, is actually having had a previous episode of depression. If you've had depression before, you're very highly likely to experience it again. So one of the first studies that we did is we wanted to look inside the human brain and see if people who are at risk of depression, i.e. people who are now considered recovered, but who have had depression in the past, how does their brain respond to reward? If reward function really is a biomarker, they might still have differences in how their brain responds to reward. And this would be similar to what the studies have already shown in depressed patients. But this was the first study that ever looked at how people who are recovered from depression responded to reward in their brain. So how do we do that? Well, it's really simple, isn't it? Humans, you just have to give them chocolate. So all you have to do is you put people in the scanner and you give them chocolate. You give them pictures of chocolate and tastes of chocolate, and they're very happy. So this is what we did. We can give tastes and pictures of chocolate, and we can deliver them directly into the scanner. Yeah, sign up at the end. We've got lots of studies for you to do. We never have a problem people signing up. So what did we find? One of the main results, and I won't go through all the results, as you can imagine, we've got quite a lot of data, but the main result to show you is that these are parts of the reward system on the left. This is the anterior cingulate, parts of the striatum. These are parts of the brain that we know are heavily innervated by the dopamine neurotransmitter, parts of the brain that we know to follow how pleasurable you find certain situations. And we found that when you put people into the scanner that are both controls, i.e. have never had a history of depression, and people who have experienced depression in the past, you get big differences in this part of the brain when you give them chocolate. So even though we asked everybody before they go in, how much do you like chocolate? How much do you eat chocolate? And they all said the same and everybody loved it. But when you put them in and you look at their brain, people who used to have depression in the past had a much smaller response to the chocolate. And this was similar to what you find in depressed patients. And so it made us think, oh, okay, maybe how you respond to reward really is a biomarker for depression, but this is just the first step. As I've probably already mentioned, these results show that even though you've had depression in the past, you still have a reduced ability to respond in your brain to reward if you're recovered from depression. It helps us think that reward function might possibly be a biomarker. And more than that, it even indicates a residual symptom that hasn't been treated. Why are people who have been recovered from depression still got differences in how they respond to reward? Is that one of the reasons why they then go on to develop episodes of depression in the future? Is this part of the risk factor? These are the kind of things that we're interested in. So you may have noticed a problem with this study, and that's okay. There's no such thing as the perfect study, as I like to tell my PhD students all the time. There's always something wrong. And the problem with this study is that we weren't sure if these differences that we found in the brain were actually real biomarkers, i.e. trait markers, just means the same thing. Risk factor, trait marker, biomarker, vulnerability, they all mean the same thing. Is it, or is it because the person had depression in the past? Is it because there's something left over? Once you've experienced depression, your brain changes. So we couldn't tell from this study. So that led us to think, well, we need to look at another at-risk group. Now we need to look at people who are also at risk of depression, but haven't yet had any scars of the illness, haven't experienced depression themselves. And so what we did is we took people who had a family history of depression. So I don't know if you know this, but if you have a parent with depression, you're approximately 40% more likely to experience depression yourself. Why is that? We don't know. We don't know if it's completely genetic or environmental. It's more likely an interaction between the two. But we do know that you're at a higher risk of experiencing depression. So the next study that we did is we took a sample of young people with a parent with depression. Their ages were 16 to 21. And what we did is we actually screened to make sure that they had a parent with depression, but they had no personal experience of depression yet. And we compared them to a sample of young people with no parent with depression. So what we would call a low-risk group. And what we hypothesized, again, if we were to really believe that reward function is a possible biomarker for depression, that there might be, again, differences in how these young people respond to reward in their brain. And what did we find? Indeed, there was. So, again, these are healthy controls on the left, and these are people who have got a parent with depression. You put them in the scanner and you give them chocolate. Again, outside the scanner, they say, I love chocolate, everything's great. They have no symptoms of depression. They don't score any differently in any questionnaire, so nothing behavioural. There's nothing we can detect subjectively, but objectively, or with a brain scanner, you can see differences in how these people are responding to reward. And so, again, this led us to believe, maybe reward function really is a biomarker, maybe it is a biological risk factor that we should be thinking more about, that we should be targeting. The particular brain area that I highlighted here is called the anterior cingulate cortex. Don't worry about too much what it does. I'll tell you what it does. It's involved in decision making, in the processing of rewarding information. It links outcome with action. So basically, it's a part of the brain that allows us to assimilate lots of different pieces of information and to go after our goals when we're making decisions. And what the results from this study showed us is that if young people have a problem in this part of the brain, I should say a young person with a parent with depression has a problem with using this part of the brain to respond to rewarding information, then maybe they're putting themselves into difficult situations. Maybe they're having difficulties making decisions. I mean, how we make decisions is all based on positive and negative information in our environment, right? So if you've got a problem in this part of the brain, maybe you're not making appropriate decisions or having appropriate behavioural strategies, which of course in turn will lead to another of the risk factors that we know for depression, which is negative life events. If you're not making very sensible decisions, not very good things happen, another risk factor for depression. But of course there's a problem with this study, as with all the studies. They're just bit by bit we try to build a picture and try to understand these processes. And what we found is, compared to other studies that looked at adolescents and how they processed reward, was that we didn't get overall deficits or decreases in how they processed reward. There was inklings of it. It wasn't that powerful, the result. And then we started to think, why could this be? So there was one other study, and they looked at young people who were 13 and up, and ours, if you remember, they were 16 years old, 16 to 21. So it's very possible that what we actually did was recruit a sample that was resilient to depression. Does that make sense? So basically, we selected a group of people who were at risk of depression because their parent had depression, but then we made sure that they never actually had any symptoms of depression themselves. So out of a group that was at risk, in a way, we may have even selected a subsample of that were highly resistant to depression, which is very interesting in its own right, and that's another whole PhD program. But it wasn't exactly what we went into the study thinking. And so, of course, this taught us what we should do in our next study. And so in the next study, we wanted to now look at two things. Younger people, okay, so let's not select adolescents 16 to 21 that have been specifically recruited because they haven't experienced depression. They could be a particular bunch of people that, like I say, is interested in it for a different reason. But maybe we should be looking at young people who actually have symptoms of depression. So they're not clinically diagnosed, they have no diagnosis, they're not in the mental health system, but when you give them questionnaires on how do you feel, how do you think about pleasurable experiences, how's your moods, they actually score quite highly on depression questionnaires. And so this was the next study that we did here at Reading University. So we selected 16 young people people between the ages of 13 to 18, and we compared them to young people that didn't score highly on mood questionnaires. And in this instance, we used what's called the family mood questionnaire. It's not, is it? What's it called? The Mood and Feelings Questionnaire. Thank you very much, Evelina. And we use this to differentiate between people who didn't have symptoms of depression and young people who did. And then we wanted to look inside the brain and see again, if these young people who are at risk of depression have differences in how they process reward, does that also map onto their actual feelings or their symptoms of depression? And this is what we find. So HR here just means high risk. And so what we found, again, is that in the grey, the light grey, these are young people with low symptoms, i.e. no symptoms of depression. And the dark grey is young people with symptoms of depression. You put them in the scanner, and again, you give them chocolate. And similarly, you find parts of the anterior cingulate decreased or not as responsive as young people with no symptoms of depression. And what was more interesting as well from this study is that because these young people actually had symptoms of depression, we could now also look at the relationship between the personal experience or the feelings of depression and the brain activity. Because of course, one of the criticisms from this type of work, from looking at pure neurobiology and no behavior, is, well, where's the relationship? Does the biology matter? If the person is or isn't experiencing the situation or experiencing the anhedonia, what does it matter? But this showed us that it really does matter. So what this graph shows, in the white squares and the flat line shows young people who have a low risk of depression, so they have low scores on depression questionnaires. There's no relationship between their brain function to reward and their how they respond on questionnaires of depression. But if you take the high risk group, they have increase in scores of anhedonia. So the more anhedonic they felt, the more depressed they felt, the lower brain activity they had in response to chocolate in the scanner. And so for us, this was a really nice study showing that you can start to link the biology, the risk factors, the biomarkers possibly, with the actual behavior or the symptoms of depression in these young people. I've probably already summarized this. So what does this mean? Adolescents with depression symptoms do indeed have responses that are reduced to reward. This reduced brain response to reward is also related to the symptom of anhedonia in this group. And this further supports our quest in trying to find neural responses to reward as a possible biomarker for depression. Of course, there's a problem. It's not a massive problem this time, but it's still a problem. We still want to know, how would young people, though, who are clinically diagnosed with depression, how would they fare in our model? How would their brains respond to reward, in particular our tasks? So when we give them chocolate and we actually give them unpleasant stimuli as well, we still don't know how that might differ. And what we suspect from the study that we're currently running right now, what we suspect is that it'll be similar to the high-risk group that I've just talked about, but actually there will be this clear correlation between symptoms. So the more severe the young person's depression and anhedonic symptoms are, the greater the deficits in how they process reward and information in the brain. So this study is just currently running at the moment here in our group. And so to summarize from this first section of the work, I just want to hopefully impart to you that we think biomarkers are important to find, so that one day it might even be possible to try and intervene early, to have early interventions in young people who we know are at risk of depression. Because unfortunately, if you remember the slide on risk factors, one of the biggest risk factors is having had an experience of depression in the past. So if we can get in there early and prevent people from actually experiencing depression, we might have a better way of preventing full-blown clinical depression in the future. Furthermore, identifying the neurobiology of depression, which is what we're interested in doing, will also help us really target our treatments. Unfortunately, the treatments that we have today are quite blanket, i.e. there's same treatments, both psychological and pharmacological, are used for many different psychiatric disorders. They're not specific to any one particular subtype of depression. They're not even really targeted to particular symptoms of depression at this stage. And this is really the goal of our research in our lab. We're really interested in trying to figure out what type of depression you might have, and if there's a particular way in which your depression could be better targeted by the treatments that we have, or even a development of a new treatment for that specific subtype. I'm going to move on now to a little bit of work that we've done as well in relation to pharmacological treatments that are used for depression. Again, we're interested in how pharmacological treatments interact with the reward system. And as you may know, and I had this in an earlier slide, that the pharmacological and psychological treatments that we have for depression are somewhat effective. They're effective for some people and work really, really well, but not for everybody. And why is that? Is it possible that some of the symptoms, some of the key features of depression just are not being targeted by the treatments that we have? For example, the psychological and actually both psychological and pharmacological treatments are not really designed to tackle the second of the two main diagnostic criteria for depression, anhedonia. And so, antidepressants. You all have heard about antidepressants. They're in the news quite a bit about how they do or they don't work. Unfortunately, they're not terribly effective, but what we do know is the ones that seem to work best, the ones that we have at the minute, are the commonly used SSRIs, selective serotonin reuptake inhibitors. And if you remember from one of my previous slides, we know that depression is maintained by a negative bias, and we know that negative processing seems to be underpinned by the neurotransmitter serotonin. And studies have shown, both in animal models and early human studies, that by manipulating serotonin, we can alleviate some of the sadness and mostly the anxious and the panicky type feelings that you get comorbid with depression. And so that's why these agents are used. We know serotonin's involved in our moods and our behaviours, our sleep, our memory function, how we learn, our appetite. They're very, very important neurotransmitters. And all of these seem to go a bit wrong when you're feeling depressed. And so again, there's good reason to see why modulating serotonin has effects on alleviating some of the symptoms in depression. And again, both psychological and pharmacological treatments in depression seem to target the negative processing. So for example, in CBT, you will talk a lot about how there's too much emphasis on the over-processing of negative information and how you could maybe try and think about other situations that aren't so negative. So my point really here is that most of the treatments, both psychological and pharmacological, target negative processing. But when people are taking antidepressants, patients report what's called emotional blunting. And this is good in that, in one sense, the sadness is gone, the anxiety, the panic, but patients don't feel happy. They don't feel motivated. They don't feel like they want to get up and get on with their life. They just feel nothing. And this is what patients and psychiatrists talk about when they're talking about emotional blunting. And we're interested in this as well from a neurobiological perspective. We're wanting to know, well, if people don't feel like getting up and going, they're not motivated, they're not feeling pleasure when they're depressed, and then you give them an antidepressant like a selective serotonin reuptake inhibitor, which just increases serotonin in the system, then maybe that makes kind of sense that this drug isn't making them feel more motivated, because we know serotonin isn't directly involved in reward. We know serotonin doesn't have, in fact, if anything, when you increase serotonin, you can decrease dopamine. So this is the kind of thing we were interested in. We wanted to see how these different treatments affect the processing of rewarding information in the human brain. So one of the first studies that we did is we took a group of healthy people, not depressed patients, and there's very good reasons for that. Number one, hopefully I've already shown you that both depressed patients and people who are at risk of depression already have differences in how they process information in their brain. So if I was to then give them a drug in a study, I would find it very difficult to understand if I was looking at a drug effect, or if I was looking at a symptom effect of the depression, or a scar of the illness, for example. So if you're a psychopharmacologist like me and you just want to understand how does a drug actually work, then you start at the basics. You start at the first step, was just trying to see how this drug will interact with the reward system in healthy people first. And that's the first thing that we did. So we took a group of people and we put them on citalopram, which is one of the most widely used antidepressants today. It's a selective serotonin reuptake inhibitor, so it increases serotonin. And we put them in the scanner and we wanted to see how they responded under this drug to rewarding and negative information. And we also had a placebo control, so that means we had another group of people who got a placebo. Neither of the groups knew what drug they were on, so that's called double blind. We didn't know what drug they were on, so there was no experiment or bias. And at the end, we only unblinded to start looking at the biological or the neural data. And what we hypothesized, is in keeping with what patients report and what psychiatrists say, is that citalopram is pretty good at taking away the negativity and depression and the feelings of anxiety and low mood and sadness. So we expected that when we give this drug to healthy people, it'll probably manipulate or modulate or change how they respond to negative information. We weren't sure what it would do to reward, to be honest. I have to put my hand up. We weren't sure. We thought it probably won't have any effect on reward. But what did we find? So when we first look at something unpleasant in the scanner, and what we do actually, we can give these nasty tastes. So it's like a medicinal strawberry. It's not very nice. And we give this nasty taste in the scanner, and we give pictures of like moldy strawberries and dead flies and things. So unpleasant situations. And we deliver this to the scanner. And what we find is that parts of the brain that we know to respond to negative information, parts of the brain that process that information, under citalopram are reduced. So these are all healthy people, this is placebo, and this is citalopram. And when you're processing negative or not very nice information, citalopram reduces the brain's response to that. And that's a good thing, right? But what happens when you give citalopram and you're processing chocolate? Well, it also takes out the chocolate. So here in the cream we have placebo, a normal healthy response when you get chocolate. This is the ventral striatum, a really key part of the reward system in the human brain, heavily innervated by dopamine. And when you give citalopram, it takes it away. So even though the person doesn't report any subjective differences, they can't say to you, oh, I feel any different, because they've just had a drug. They don't say they like chocolate any different. They're not reporting wanting to eat it less. Nothing subjective. But in the brain, we find these big differences this drug is having in healthy people after only 7-day treatment of citalopram. And so what does it mean? Well, for us, we thought it was really interesting. This was one of the first studies to look at the effects of citalopram on reward in the human brain. And of course you would think, why don't people look at this? But actually you'd be surprised at how antidepressants are actually get through from clinical trials into patients. And they're pretty much, you're just asked, does it make you feel better? And if a drug makes you feel better compared to placebo, that's how they get passed. Today, there is no neurobiological data or anything like that used in a clinical trial, for example, for a new drug to meet the market, just in case you don't know that. So basically, these results show us that citalopram seems to be good at taking away the negative processing, but maybe it's not so good for positive. So it kind of fits with the story that you get from patients saying about, I feel kind of nothing, I don't feel sad anymore, but I don't really feel very happy. And so there's been reports by some of the leaders in the field, some of the leading psychopharmacologists like David Nutt, have published reports saying that from his experience as a psychiatrist and using drugs that manipulate dopamine and noradrenaline, that they seem to be better at treating patients who have dominant symptoms of anhedonia. Now again, this is just a notion, a clinical report. There's no evidence, there's no biological evidence, there's no clinical trials on it yet. And so this is the kind of thing that we're interested in. We're kind of leaning towards trying to find the evidence that certain drugs might be better for certain symptoms of depression. And so this study made us question, well, maybe drugs that manipulate dopamine, noradrenaline, maybe they're better at treating the symptoms of anhedonia. Well, first of all, how do those drugs even work in the human brain? What do they do to the reward system in the human brain? Let's have a look. So the next study that we did here at Reading is we looked at the effects of 7-day treatment with bupropion compared to placebo. Now, bupropion is a drug that blocks the reuptake of dopamine and noradrenaline in the system, so it keeps more noradrenaline and dopamine alive, so to speak. And it's a drug that's actually licensed in the US as an antidepressant and is sometimes used as an adjunct therapy, and that just means sometimes clinicians of their own accord decide, in my experience, this drug works quite well for this type of person, this particular depression subtype. I will give it alongside an SSRI. And so that's what we know about these drugs so far, that they seem to be good at treating certain symptoms of depression like anhedonia. And they don't have some of the same negative side effects that the serotonergic drugs do. But like I say, as far as how the brain actually, the drug actually interacts with the brain response, there's very, very little data. And so what we did is we took the drug again, gave it to healthy people, as we wanted to look at the mechanism of action, not a clinical response. And we gave it into our study where we look at both positive and negative information. And if we think about the positive, what did we find? Well, in comparison to citalopram, we got the complete opposite. So when you give people chocolate in the scanner and you give them 7 days of bupropion, you get a huge increased response to chocolate in the brain. So there's a potentiation of reward or an enhancement of the process and of rewarding information. And this is what we would have hypothesized. And so the results of this study just go to show that in fact, dopamine drugs or drugs that interact with the catecholamines like norepinephrine, dopamine seem to be good for maybe a lack of reward in depressed patients. So patients that maybe have dominant features of anhedonia might be better suited to these type of treatments rather than the treatments that are better suited, the serotonergic treatments are better suited for patients who have more anxiety in their depression or more processing of negative information like a comorbid panic disorder, for example. And this is what clinicians report, it's what patients report, but these are some of the very few only studies looking at how that maps onto the actual biology and the mechanism of action of these drugs in the human brain. Furthermore, just the last point here is important to note, when patients are on antidepressants and they report this emotional blunting, so this feeling of neither feeling terribly sad but not feeling very happy either, I mean, is it possible that that's the symptoms of their disorder, their depression, or it could even be a side effect of the citalopram, of the serotonergic drug? We're not even completely sure of the answer of that. And yet with the studies into these other drugs that potentiate dopamine, it's possible that they might have less side effects, this less ability to dampen down the processing of reward, considering we find that they potentiate reward or increase the processing of reward. And so I'm nearly finished actually. So this takes me to the overall summary. I hope I've been able to show you that a combination of these studies that we're able to measure how the brain responds to reward and how this is reduced in people who are at risk of depression, which is similar to what you find in depressed patients, okay? And that this reduced brain response also correlates or is related to feelings or symptoms of actual depression. So that gets around the criticism that, oh, neurobiology doesn't mean anything, well, it's all about the behavior. But if you can marry the two and you can show that actually you can detect biological differences in somebody before they even get ill, and that you know that these biological differences can predict onset, then maybe these are true biomarkers and worth targeting both as interventions and also with our current treatments, being more specific with our current treatments. So the last point here I'd like to get across really is that yes, psychological and pharmacological treatments work. They work for some people and they work well for some people, and they focus mostly on negative processing, and that's great. But maybe we should start thinking as well about reward processing and some of the other major symptoms of depression. And if we're not targeting those with both our psychological and pharmacological treatments, maybe this is why certain people relapse or never fully recover from depression. So this just takes me to thank a lot of the people that's been involved in this work. In particular for the early studies is when I was at Oxford University, and now some of the PhD students that have been involved in the at-risk studies, Evelina and Jennifer, and Zola, who's been involved in the drug studies at Reading, and some of our medics as well, who've been really, really beneficial in carrying out our drug studies. Stephanie Horndash, who's gone back to Germany, and Panos Giannopoulos, who would like to thank them as well. And this just leaves me, finally, to thank you very, very much for listening. And I'll take some questions if you have any.